Sir, we just give a short introduction, sir, then you can share your screen, sir.
Right. So,
Ma'am.
Yeah, please, Sindhu.
So, good evening, everyone. We welcome you back to our Advances in Embryology Masterclass series. It is truly an honor to have a very experienced and dynamic Dr. Sanjay Shukla, sir, with us today. So, before we introduce sir, we'd just like to inform you that, as you all know, mostly that we have two certificate courses now. One is Genetics and Infertility, which is a 12 weeks online course. We've successfully completed two sessions already, and it's an ongoing course. And the other course is reproductive immunology, which is an 8 weeks online course again with once a week classes. It's going to begin next week. We already have a lot of registrants, and it is definitely going to be useful for anybody in embryology and infertility. So with that, I have the privilege to introduce Sanjay Shukla, sir, who's the clinical embryologist at Asta Ferticare Jaipur, Diva Women Care and IVF Center Jaipur, Shivani IVF and Fertility Center Jaipur. He's working in the field of human reproduction since 1998, since most of us were kids. And he has a PhD in life sciences, and he's worked on the early diagnosis of typhoid fever. And he has a fellowship in biology of reproduction from Argentina, and he was the past president of ACE, and he's a former adjunct professor and senior consultant at Mahatma Gandhi University of Medical Sciences and Technology, Jaipur. Sir is well known to many of us, all of us, and he's an icon in embryology. So we're very grateful that you accepted our invitation. And you're here with us this evening, despite your busy schedule. We know you would have just reached home. This shows your passion for teaching, sir. So thank you so much.
Good evening. Again, I would extend my sincere gratitude for Dr. Sanjay Shukla sir for, taking this platform and, addressing all of us. And today he's going to talk about the embryo culture media, our day-to-day, life in the lab. None other than him can do justice, because I think since 1998 is, I don't know whether he has made his own culture media. We are all working with the recipes, which is, ready made and, very easy for us to use, whether it's single step or, sequential. Many things are so important, the ingredients into it, the nutrients into it, which is required for which stage of embryo development, and how it is going to, make a cleavage stage into a blastocyst. Everything depends on the contents of the culture media. And the meticulous composition not only nurtures the embryo, but it also has an impact on the embryo quality and how you make these culture dishes and so on and so forth. So it is definitely a critical lifeline of any embryology lab. And without taking much of your time, I think I would request Dr. Sanjay sir to please take over this session. And we are very eager to listen to you.
so at the outset, I would like to thank Dr. Vasundara and Dr. Sindhuja both. Um, Actually, I must apologize them because it was earlier given to me long back, and I was expected to to take this opportunity. Uh, Incidentally, as you mentioned, there were some other commitments, and you know, because these days I'm working as a freelancer, and that makes a life bit complicated for me. And of course, the mitochondria are taking over now. So, uh, uh, anyways, uh, I'm thankful that you accepted my apologies and rescheduled the so-called master class. I don't consider myself as a master. I believe I'm still learning, and also I think this particular session, most of the attendees are the ones who are either students or the juniors or the beginners in the field of embryology. So I'll try to keep it as simple as possible, because this topic is extremely overwhelming even for me. It's very complicated, and the masters have written books on this particular topic, so it is not possible to accommodate entire thing in just one lecture. Or So what I'll do is I'll give you an overview and then try to convey some of the practical tips and the things that are essential in day-to-day working of an embryologist. So hope uh, I can justify the responsibility given to me.
A lot of
Uh,
clinicians have also joined, sir. We are eager to listen to you as well. We want to learn more about culture media and how the lab functions. So we are very,
And
very interested.
also, as you mentioned, yes, I do have hands-on experience with cooking my own media. Not just, I mean, before entering into embryology, those were the days when, as such, the embryology fraternity was not developed in our country. And as you mentioned in my CV slide also, that I belong to microbiology field, and it was a premier institute in central India where I was doing my PhD those days, working on the Salmonella typhi, and there were. I would like to, if I'm allowed to, I'll give a little brief about how I started, because probably that can motivate the students and they can also think of how life has become easier nowadays. Uh, so we were six PhD students and working on different um, subjects in the uh, microbiology, immunology, serology, partly plant uh, biotechnology also. So uh, those were the days when we had limited resources and we used to prepare everything at our own. We were fortunate that the uh, owner of the hospital gave us a free hand, so whatever we wanted we could ask them and they would provide us. But that experience has given us an immense depth of understanding of biotechnology and also different aspects of clinical application of research and basic science into the clinical settings. So that was a very enriching experience for me. And that helped me when I uh, migrated from clinical microbiology laboratory to clinical embryology. Uh, but at that time, when I started, we were getting ready-made media. But when I joined another laboratory, it was designed well, uh, you know, back when there were some shortages of media, shortage of equipment. There was a ban from importing the reagents and equipment and all. So there was a period when you recruit the patients and then you do not have the medium, because at the time that media you order from Sydney or somewhere, and then by the time it reaches you, your patient is not there for IVF. So it was, an sort of, emergency or whatever you say. Uh, so we were obliged to prepare some sort of media and did some experiments. We had this osmometer to check all the osmolality of the media, pH meters, scale, then reagents, the filtration, everything. So it was a very tedious job. And ultimately, whatever we have done, it was suboptimal. So now these commercially available media, so I wonder that uh, they are, you know they have made a tremendous advancement and progress in the quality of the media. And what we were achieving at that time, 15 to 20% success, now we have crossed 50%. So it is a substantial advancement. And um, I must thank all the pioneers in the field who have not just helped us to achieve this landmark of more than 50%, which is even not there in natural reproduction. So, and I'm also fortunate to have interacted with some of the pioneers during uh, my journey. So, okay, let me start sharing my um, slide. Uh, so, uh, just 1 second. Is it visible?
Screen is visible, sir. It's not in slideshow yet.
Okay, okay. Let me go back.
Any difficulty, sir?
Not exactly. Is it okay?
So we are seeing it in the presenter mode actually. So we can see the previous slide, next slide, notes.
Okay.
Whatever you are seeing, I think we can see.
Um, in a moment, just give me because
Sure, sir. No problem, sir. Take
yeah,
it.
yeah.
Perfect, sir.
Is it okay now?
Yes, sir.
All right. So, uh, this you have already taken care of, so I must skip this. Uh, so it's very beautiful uh, statement from Dr. Thompson, uh, where he writes that there is no adaptation by an embryo to its environment that has no consequence. And we, as a scientist, must all remember, because whatever we do in our laboratory with the gametes or the embryos has its consequences, not just during the embryogenesis in our laboratory, but far beyond that. I mean, it may manifest even in the later stages of that particular individual if the embryo is conceived and the uh, baby is born. So we must be very responsibly work in our laboratories, and like there are several uh, articles and several guidelines in the um, published literature that say that everything is important in our laboratory, whatever we do. So be responsible whenever we start doing any clinical work in IVF laboratory. There is a testing time when we transit from the trainees or junior embryologist to a person who is actually working in the laboratory as a senior embryologist or embryologist. So we have immense responsibility over our shoulders so that we can deliver the best to our patients, and uh, do whatever is best to achieve a healthy pregnancy, not just the pregnancy, a healthy uh, baby that can be, that can uh, live a healthy life uh, even after the birth of the baby. So this has caught my attention, and in particular, differences in embryo culture medium composition may lead to differences in the adaptive responses to stress. We all know and probably you might have seen that particular picture in Dr. Gardner's book, where he has given the, uh, the degree of tolerance of an embryo, not just an embryo, from egg to blastocyst, that it increases the tolerance limit when it progresses to blastocyst. But that limit, as we say it, to the embryoplasticity has its own consequences. So we just not take it for granted that embryos can adapt to anything. So be responsible. And with that mindset, if you work in the laboratory, you can produce better results. So, first, let us uh, take a little dive into the uh, development of the uh, or the evolution of human embryo culture media. So, uh, it started not just uh, recently, actually, maybe 100 years back, when the scientists started working on different you know uh, aspects of reproduction in animals, and they tried to achieve fertilization or the embryo development in different animal species. So from there, this progress, as I mentioned earlier, has advanced substantially. And as the uh, uh, understanding grew, so we started optimizing the embryo culture, not just embryo culture medium, but also the embryo culture conditions. And the growth of ART has seen those golden days where we are working now. So this is a timeline. We can divide it maybe in four categories. So time before 19, yeah, or uh, up to 1950s and 60s, then from 60s to 80s, then 90s to 2000s, and the present day embryo culture. Okay, so it all started with these simple saline solutions. For example, Ringer's solution. So earlier, uh, what we knew that the biological fluid, or maybe you can say the human blood, is considered as the gold standard to prepare these solutions where we can keep our uh, tissue or cells to study. And it was adopted that, okay, for example, a simple saline, we'll put everything in this and observe the cells or tissue or their metabolic activities or their growth. So we had this simple thing for many years. So and this the saline solutions or the so called medium were not actually the medium, but uh, a simple salt solution that can help keep the cells or cell lines or tissue viable for some time. So these were not actually the culture medium in true sense, but just the support medium. And therefore, as the experiments uh, advanced, people realized that this is not sufficient. We need to improve on the uh, things to meet the requirements, and therefore the uh, the field advanced in later years. So, uh, in 56, scientist named Witten, he developed the first culture medium that was completely defined for culturing the mouse embryos. Of course, it was based on the Krebs ringus bicarbonate, which we call KRB. This had been initially formulated in 1932 by Dr. Krebs. Mouse embryo did not grow in KRB. So Witten's medium differed from this KRB only by the addition of uh, sugar like glucose and BSA, bovine serum albumin. And this modification supported the development from 8 cell of the mouse embryos through blastocyst stages. So this was a very significant uh, change from what we are using, the simple salt solutions, to this uh, glucose and BSA added medium. So Dr. Witten was probably the first to grow the mouse embryos up to blastocyst. So here is a timeline. If you see 1932, then 1956, he added glucose and BSA. Later, they also added lactate because these embryos earlier in their experiment, they grew them from 8 cell because what they did, those who have worked in the animal laboratories, they probably know that the oocytes from or the embryos from mouse are retrieved by flushing the oviducts and then retrieving the uh, already fertilized embryos. So they grew those 8 cell embryos to blastocyst, but they could not grow 2 cell embryos or zygotes. So they added lactate. Now, this lactate helped them to grow the mouse embryos from 2 cell onwards, but again, they could not grow zygotes, the fertilized eggs. And this remained a puzzle for years. In 1967, Biggers, Whittington, and Donahue, they added pyruvate to this medium and they overcame this problem of growing the mouse embryos from 2 cell to further up. And this was named as M16 medium. Now, what is this 2 cell block? Probably we will also discuss this in later or this talk, because this was observed by many other people. Then see, from 67, I mean in 70s to 90s, there was not much advancement. Then this particular medium, CZB, and another medium, KSOM, were developed in the early 90s. CZB stands for the initials of the scientists Chatot, Ziomek, and Bavister. So this medium was modification in M16. What they did, they added glutamine to it. They also added EDTA, and they improved the lactate to pyruvate ratio in this. So, and this was, this CZB medium was two-step medium. So first 2 days, there was no glucose, and after 2 days, they added glucose. And this helped them to overcome this two-cell block, and they could culture the embryos, mouse embryos, up to blastocyst. Dr. Biggers developed KSOM. Here, SOM stands for the optimizing medium, simplex optimization medium. And you can understand by the word optimization that they probably used some algorithm, trial and error, that okay, let us add this ingredient, or let us reduce the concentration of this one. And so they optimized with different trial and errors, of course, based on scientific understandings. So they developed this KSOM. First the SOM, and in this they use the glucose throughout, not, I mean, like the CZB medium, uh, there was no glucose in first 2 days, but they used glucose throughout, and still they could achieve blastocyst from zygotes. This K word is this K word indicates the potassium. So they also realize that increasing potassium, or adding potassium to this simplex optimized medium, helps grow that, or overcome the two-cell block, and helps the uh, embryos to grow up to blastocyst, even if there is glucose from day one. Now in 90s, again, Dr. Bevister, okay, let me tell you a little about Dr. Bevister. We owe to him a lot. He was one of the colleagues of great Professor Edwards. He was working in his laboratory, not his laboratory, but one of his friends' laboratory on hamster oocytes. And he was the one who advised Dr. Edwards that, because at that time they were having very, poor fertilization. So he advised, if we change the pH, we can uh, achieve better fertilization. So of course, we all remember what Professor Edwards and Jean Purdy and Professor Dr. Steptoe has done. But even Dr. Bevister's contribution to first uh, success of IVF cannot be underscored. So Dr. Bevister found that some amino acids are good for the mouse embryo culture and some are not. Now, later we all know what Dr. Gardner and Mitchell Lane has done. Uh, they also working on similar lines, and they found that non-essential amino acids are needed for cleavage stage embryos, and non-essential amino acids as well as the essential amino acids are needed for later stage embryos. So these are the landmarks in the history. I have simplified it. I mean, there are so many other contributors to this knowledge, but I have just simplified it so that we can acknowledge what the pioneers have done. And you can, appreciate from early 90s to 2025 what we have achieved. And with those, limited resources, what they have done. So this is remarkable. Then in 70s, you all know what 70s has brought to us, the uh, birth of first, or the conception of first IVF baby. And then uh, so from simple uh, medium, simple modified saline salt solutions, and then addition of different you know ingredients to it, we came to complex tissue culture media, because we realized that the blood is not an ideal uh, model for the cell culture. So a lot of things changed during this period, and we learn that uh, the embryos, even the embryos, are different from the other tissue or cells that are being cultured in different media. So we need embryo specific media. So during 1970s to 80s, people realized this. This led to the introduction of amino acids as suggested by Webster and Dr. Gardner, some vitamins, and serum, so that we can culture the embryo specifically in this medium. So the basic uh, the base medium was hemceftem, uh, which Dr. Edwards used, and it included glucose, lactate, pyruvate, amino acids, and this resulted in the first successful human IVF, and Louis Brown was conceived and born. Those who have seen the film Joy must have uh, realized how difficult the feat was. So there were challenges with this. Uh, Despite this progress, serum introduced batch variability, because every time, even from the same individual, the serum will differ in its constituents, and again it poses the risk of uh, infections or the immune reactions. So this medium was also suboptimum, and therefore during early days of IVF, even when I started working, so we were doing mostly day 2 transfers. Sometimes if we left the supernumerary, because those days we did not have that robust cryopreservation system in our laboratories. So sometimes we used to leave the embryos in the um, medium itself, and at that time it was single, not sequential medium, but a single medium, IVF medium, what we used to call it. And even we could see some blastocysts growing in it, but that was very suboptimum culture condition. So not many blastocysts were developing in this medium. So mostly we were transferring day 2 or day 3 embryos. Then came the era when Dr. Gardner started um, developing his sequential media. and it revolutionized the human IVF entirely, because he understood that the developing embryos need different uh, nutrients, or their metabolic needs are different at different stages, starting from the zygote to blastocyst. So he uh, developed G series media, G1, G2, what we say, and of course, Dr. Quinn was also, Patrick Quinn was also working on this in the same line. So he also developed uh, media. It was earlier known as Quinn's medium. So, and then there were some other similar media available, commercially available, so that we could culture uh, embryos to blastocyst successfully. And this is in humans, right? So we were talking about mouse embryos earlier, but then we translated that understanding or that information or that knowledge that we gained from the earlier experiments from the mouse, the cattles, rabbits, hamsters into clinical um, human application once the Louis Brown was born. So it had significant impact, because then every laboratory worldwide started uh, culturing embryos up to blastocyst. Why blastocyst? Because we knew that this is the stage when the embryos come into the uterus in a natural cycle. So we always try to mimic what happens in nature. And once we started culturing embryos up to blastocyst, our results started improving day by day. Then, as mentioned earlier, the protein source was also a problem, because we were using in the olden days the serum, then some purified proteins, but those proteins or the serum are um, the biological sources, and they had the risk of transferring not just the pathogens, the viruses, the bacteria, or the other particles, but also there was a substantial uh, variability in the composition. So people started acknowledging that we need some other source. So why don't we uh, define our media and keep them protein free? In fact, there was a time when we were receiving the commercially available media without protein, and they were also supplying us protein in the form of albumin, and we used to add protein just before culturing our embryos, or just before starting our IVF, um, you know, uh, dish preparation the day before the OPU. So, of course, this again improved the things, but again, problem that it was tedious thing, and there were certain other issues. So, people started to work on some synthetic protein. So later stages, what we are using now, they are not exactly the uh, you know albumin that are coming from the animal sources or human sources, but they are mostly, I mean, there may be human source, but they are either recombinant or the synthetic serum supplements. So this has also changed, and this has also, I mean, this was important because there were certain legal complications. So to meet those regular uh, um, regulations and compliance, especially in European Union, you needed to switch from the uh, bovine serum albumin or human serum albumin to recombinant ones or the synthetic serum supplement or synthetic protein supplements. And then 2010, so this was the next gen medium. Uh, we saw the advent of time-lapse technology, and this time-lapse technology um, prompted the scientists to develop medium so that we can keep the embryos in culture undisturbed. So we needed an uninterrupted uh, culture system. And for that, they started working on the single step media. So as you know, many people like Jacques Cohen and others, they used to say, uh, let the embryos choose. So this theory worked in favor of the single step media, and they added everything which is required by the embryo, starting from the fertilization to blastocyst in just one medium. And this continuous culture medium helped people grow the embryos without changing the medium. And probably this has become the most popular uh, style or popular culture system uh, all over the world. And most of our laboratories now are using mostly single step media. There are few people who are still, they are um, admirer of the sequential culture medium, but most of there are certain other aspect of also that has helped popularizing the single step medium. Also, uh, remarkable work again from Dr. Gardner and some other researchers who they said that if you need to culture blastocyst, the physiological oxygen should be there in your uh, culture system. Earlier, we were using the box type of incubators where we had the uh, ambient air, and this had 21% oxygen. So we were just supplying CO2 to our incubators, and that was maintaining maybe 5 to 6% CO2, but the oxygen pressure, oxygen concentration was 21%, which is the ambient oxygen concentration. So they recommended to use the 5% oxygen, which is physiological oxygen, which is there in the genital tract. And this again improved because earlier what we had when we started using sequential culture media, maybe 20 to 30% blastocysts. But now we have surpassed even, you know, in good laboratories, consistently they are achieving 70% or more blastocysts. Not everyone. So even if you are achieving more than 50% or more than 40% blastocysts, it is good enough. But those which are consistently good laboratories, they are achieving maybe more than 70% blastocysts. So in conjunction with the time lapse, uninterrupted culture system, and this physiological oxygen, um, we have improved our blastocyst culture a lot. Now the things are coming up like the microfluidic system. We are already using microfluidics for sperm preparation, but there are microfluidic systems in offing for embryo culture. And I know one of the pioneers whom I interacted, Dr. Gary D. Smith. So he's working on this concept of lab on chip, where you can just uh, introduce the sperms, the prepared sperms, and the eggs and rest of the things, starting from the fertilization to blastocyst, will be done in that microfluidic system itself without any intervention of the human beings. You will have to add probably different um, reagents, or you can say media, and the media composition will also alter according to the need of the embryo. Say if you need, you know, less glucose during the first few days, it will be an environment where you will have less glucose, and later on, as the embryo uh, progresses from the uh, micro channels and reaches further up, the the culture milieu changes according to the need of the embryo. So this is fascinating, and probably uh, in coming 5 to 10 years, we may have a system like microfluidic system where we'll just uh, put the gametes and sit back. Then xeno-free and chemically defined, like what we have, as I mentioned, the especially the macromolecules, the albumin supplements, they have certain um, foreign uh, uh, ingredients also which are not needed there. So the supplements will be made xeno-free, and we'll have a very defined, very synthetic uh, synthetic uh, media, which is more uh, you know mimics more as the natural environment. Fascinating, isn't it? So, okay. So this was, you know, a little about the how the pioneers have worked and designed and defined the media over the years. So let me tell you, I'm not going into the details of this because this is a very vast topic. So there are certain essential components of embryo culture media. Inorganic salts. Uh, Basically, there are 6 ions, you know, in all culture media. They are sodium, potassium, chloride, calcium, magnesium, and sulfate. So you will find these 6 ions in all the media, all the culture media, and some also have this phosphate, but not all the media. So these 6 ions are there in all the media, and they are the main components, because how we started from the KRB, the simple saline, and saline was nothing but the uh, salt solution in water. Now we have the amino acids. What they do, they are the building blocks for proteins. So the fundamental role of amino acids is as the precursor of protein synthesis, and they are vital for cell structure, function, and growth. They also act as energy substrates. So specific amino acids like this glutamine, it can be used as intermediary of metabolism and as energy source for the energy ATP production. Then osmoregulation, they maintain the osmolarity. They also help in pH buffering. So certain amino acids, they help. They act as the osmolytes and maintain the cellular volume. They also help in buffering the intracellular pH. And this will, help in protecting the embryos from stressors. They also act as antioxidants. So there are various other roles, and uh, there are other constituents, but I'm not mentioning because uh, they are, you know, there is a plethora of literature where you can study those. I'm showing you this particular picture. You already know this. We have already discussed about these things. But this is the fundamental of the, the modern day embryo culture or embryo culture conditions, embryo culture systems. And what this picture depicts, that what we need to do when we culture the embryos, we need to mimic what happens in the nature. So if you see the fimbrial part of the fallopian tube, where the egg comes and meets the sperm, has a different milieu, and this environment, these conditions of the nutrients and substrates and all other things keep on changing as this particular uh, oocyte or zygote moves ahead towards the uterus. So it is not, you know, a static condition. It is a dynamic thing. It changes. I consider it as our country. Every 50 kilometers, the things change. So similarly, even the oviductal fluid is not the same in the fimbrial area and the cornual area. So it keeps on changing. And therefore, we need to work on these lines, that the embryos, to give them the optimum thing for culture, we need to mimic what happens in the nature. And uh, I must recommend you to read the physiology and the metabolism of embryos. Uh, there are several, milestone publications, but if you cannot get those, at least read Dr. Gardner's book. Uh, I believe this is even, more than enough to learn from. So we also need now. Learning from these the earlier picture, we now know we can divide these things, that pre-compaction uh, needs of embryos are different, and post-compaction needs of embryos are different. I mentioned about the two-cell block of mouse embryos earlier. Similar thing was observed by many other workers, that in different species, the embryos have some sort of, cell block, maybe two cell block, four cell block, eight cell block, and they arrest there and they do not grow further to blastocyst. So people realize that this is the time when actually the embryonic genome activates, and therefore there is something that inhibits the embryos to grow further. So all the focus was on finding those things, those factors, and then we realized that, yeah, the needs are different. The concentration of the glucose, or the other energy sources like pyruvate or the um, lactate, is different in different stage embryos. And once we learned that, we overcame the those two cell, four cell, eight cell block, and we achieved the blastocyst. There are several uh, publications regarding this. Was all about the culture media, and how uh, we I mean the scientists, the pioneers, they defined and worked on those uh, culture media preparations and formulations, and have reached up to this stage what we are using today. But then there are several publications regarding the effect of culture medium on the health of offsprings or embryos. So, the publications or the literatures is flooded with such studies where they have shown the concern that, yeah, the media, culture media, have their impact on the health of the embryos or the fetuses or the babies. So these are some of them. Um, some studied that culture media can affect the growth, can affect the weight of the baby. It may affect the cardiovascular uh, health of the children. And some, I mean, there are, like any other publications, any other studies in the reproductive medicine, these studies also suffer uh, design biases and other bias, uh, and also because we do not have a luxury to study different aspects in clinical settings. Most of the things are actually the retrospective studies. So um, they have certain limitations, but still the the concern remains that the uh, babies born uh, with the help of ARTs are prone to uh, many health conditions compared to their uh, normally conceived counterparts. Then there are some other publications like this one. You probably know there are certain very big names of modern days. Uh, Professor Anne Sunday, Joyce Harper, Daniel Brisson, Christina Magli. So it was published in 2016, posing this question whether we should take the, or it's time to, I mean, it's not questioning, it's suggestions and it's a recommendation that this is the time to take the human culture seriously. How? And they studied many other publications by that time, and they summarized in this paper, and I read out those recommendations for you, that the glucose inhibition of embryo development is still a matter of debate. What we learned earlier in the mouse experiments, that if we added glucose, the glucose inhibited further growth. So it is still a matter of debate, but it is now generally accepted that glucose should be included in media, but the concentration that we found in the human oviduct or the fallopian tube. The importance of amino acids is attributed to their roles in protein and protein synthesis, acting as osmolytes and a precursor for signaling molecules, substrate for energy metabolism. No currently used media at this time, 2016, contain optimal concentration of amino acids as they suggested. And probably it stands true even today. Then glutamine. It was a, a matter of concern, because probably you might have read it or you might have heard it from your seniors, that earlier it was a concern that the embryo culture drops, they accumulate ammonium, or there is a buildup of ammonium in the embryo culture dishes, and this buildup of ammonium is toxic, so it is not good. Then it was uh, realized that yes, it happens, but if we replace this glutamine with a more stable uh, form of glutamine, what we call is a dipeptide glutamine, so it slows down the breakdown of the products, and the accumulation of ammonium or generation of ammonium is uh, substantially reduced. Then the EDTA supplementation. You know what the EDTA does is it chelates to uh, various molecules, and specifically it chelates the uh, you know uh, iron containing things, the other macromolecules, the substrates and the toxins. It also regulates the glycolysis. So um, this was also um, you know a matter of debate whether we should in, uh, include in the medium or not. But then this was realized that the lower concentration of EDTA uh, do not support this statement that uh, we need EDTA in higher concentration, because the same uh, effect was achieved by adding lower concentrations also. Uh, Same with which we discussed about the heterogeneity of human serum albumin. So it needs to be controlled. There were certain reports when there was a problem with the human serum albumin, and then the manufacturers had to recall those batches. So um, after the introduction of recombinant HSA and uh, synthetic serum supplements, this problem has taken care of. Then addition of uh, hyaluron uh, is advantageous, and uh, but whether it should be in all the media or just in the embryo transfer medium, it is still uh, not clear. There are some certain other recommendations regarding the growth factors and cytokines and other things. So uh, I recommend you to please go through this, and it is very pertinent to read such remarkable papers. Our friend Dean Morbeck, he was the one who actually analyzed the available commercial media. Before this, uh, believe me, we were all believing that whatever the manufacturer are saying, their medium has that particular ingredient. But it was, you know, we can say blind faith. And these manufacturers, because of their commercial interest, would never disclose the concentration of the ingredients. And in fact, some of the ingredients are even not, uh, you know, made public. So he was the one, or his team was the first who actually analyzed the commercially available media, and he revealed the composition of those media at that time. It was published in Fertility Sterility in 2014. And again in 2017, when the single step media was introduced, they again did the similar experiment with the uh, murine embryos, and they found that the culture media composition varies widely, with differences in pyruvate, lactate, um, and amino acids were especially notable. The blastocyst development was culture media dependent, and it showed in interaction with oxygen concentration and presence of protein. which was, of course, already known to some extent. They also compared, four single step culture media which were there in 2017, and they found the similar uh, things like earlier, that there was a notable variation in the composition, and therefore uh, people started asking the uh, companies, the manufacturers, that they should actually give us, the end users, the consumer of their products, the actual concentration and composition of media, because this has a direct impact on the uh, outcomes and the health of babies. This is a very good editorial, uh, again published in Fertility Sterility in 23, and Professor Pawson asked, because um, he was discussing with his uh, colleagues and some uh, junior in his laboratory, and he was surprised that they were not aware of what media contains. So he posted this editorial in Fertility Sterility, and it's very, you know, striking question. What do you mean? You don't know what is in the culture media? And that's what we are discussing today. Even today, we do not know. So he reiterates what Dean Morbeck has, raised, that we should now ask the manufacturers what is the composition. Now, this is a latest publication, 2025, similar thing which Dean Morbeck and his group has done. And these people have also, compared the composition of commercially available human culture media. And again, the similar findings, that they vary remarkably. Of course, there are certain things which have, which are close to what we found in the uh, genital tract, but still there are certain elements in the uh, culture media that differ notably from one manufacturer. Not just one manufacturer, even there are parent companies which produce different brands of media and they differ. So, uh, of course, as I mentioned, that human embryos have a plasticity, they can adapt to different conditions, but this is not optimum. You are you are exposing them to stresses, and every stress has its own impact on the uh, embryos and um, growing babies. Okay, so learning all these things, uh, still, having said and read and understood all this, a pertinent and important questions remains unanswered. And this question, whenever I go in a classroom or in a meeting or a CME or conference, people ask, which medium should I use in my lab? Now, this is an eternal dilemma. We have commercial media available, and because of certain regulations, certain legal complexities, now we cannot prepare media in our laboratories at our own. We are not equipped. We do not know enough to you know cook the broth. But whatever is available, of course, we should acknowledge and respect that the people who have devised these media have given their lives. And we just can't say, what we do is we discuss with our friends, our colleagues, our peers, अरे तुम कौन सा मीडियम यूज करते हो, what do you use? And he or she would give you the experience that they have in their laboratories. And based on that experience or observations, they decide, oh, no, no, brand A is not good. I'm getting better results with brand B. But it's very vague statement and it's not a scientific statement. So what I suggest, whatever medium you have, even, we must realize that even if we have a similar sort of laboratory designed by same um, you know architect, having similar brands of equipment in our laboratory, using the similar brand of media, we are not the same. It's different. Even the same woman does not produce similar quality eggs or gametes, or a man does not produce same kind of sperm every time. They differ. So every IVF cycle is unique, and so does your laboratory. So whatever medium you choose, you should optimize it for your laboratory. And how you can do that? You can start with a few cases earlier and see if that works fine. Now we know there are certain quality control measures. We know the impact of air quality, we know the impact of temperature, we know the impact of osmolality, we know the impact of humidity, the oxygen concentration, everything, light. You know, we often neglect this particular aspect, but even the light can affect your culture conditions. So same medium may not produce the results that other one is having. So you have to work on this. Choose whatever medium you want and then see if you are doing all other things right. And for that, we need to read again that Vienna consensus document that everything matters. And we need to master those things. We need to control those things in our laboratory. This is again a humble suggestion if you want to know, because it's okay that you can select different equipment and very good, you know, uh, instruments in your laboratory. But the simple things like how to handle the media and disposables in laboratory can also make a difference. I have written this article and this chapter, and it was published in the book which was edited by Dr. Deshmukh and Chimote. So I recommend to read. I hope that the students will be benefited with this. So thank you very much. I hope uh, I have given uh, some, information from whatever little I know about the culture media and culture conditions to you. I also recommend that, um, as I mentioned in the very beginning, that I am a student and keep on reading. There are several books available. One I mentioned about the um, Dr. Gardner's book. Another book is this. I do not know whether you can see this. It
So
is
can't
it? Oh,
see
no.
clearly. Can you just focus it, sir, a little bit?
I'll try to. I'll try to. Uh, is it okay? So this is edited by Professor Patrick Quinn, the Culture Media Solutions and Systems in Human ART. Uh, I highly recommend to everyone to read this book. Uh, I happened to visit one of the manufacturers uh, factory in Denmark, and I was amazed to see how they have, you know, automated the entire process, and it is, you know, entirely human touch less conditions. So uh, it feels like you are visiting some uh, fantasy laboratory or fantasy area, sci-fi sort of thing. And I strongly believe that all media are at par. No one is superior or inferior. Maybe there are some differences, but we need to optimize our conditions and achieve whatever best we can. So this is my philosophy, and probably this should work in your laboratories also.
Thank you, sir, for that exhaustive lecture. And you've been speaking for so long, like ma'am said, please have a sip of water because we have a lineup of questions in the chat box for you.
Oh God. Okay.
So
It is
anything
so
that,
nice to hear from you because you took us through the milestones because it's very, very important. Because currently we are not taking any pains to, we are just embryologists are doing the dishes, the embryos are growing in different culture medias. You made the medias, and what difference has it made over the years? You clearly actually took us through all the events that happened. And it is very, very important for all of us to know. And despite all this, we are still evolving in terms of culture media, because so many commercialization and so many add-ons to the culture media, and it add-ons to our confusion as well. So I
True.
think basics have to be very clear. Basics don't change, but I think we have to keep evolving. Sindhu, without taking much time, can you just ask the questions on
yeah.
the chat box to sir?
So the first question is, do embryos grown in older types of culture media show any long-term differences in gene expression or development compared to those grown in modern media?
Yes, so there are studies even on the transcriptomics and other metabolomics also. And um, since they are very limited studies, so they do not have any concrete conclusion, but there are certain indications that some media types, not just the older ones, even the modern ones also, they do have some sort of impact on the gene expressions. Whether this is a negative or positive impact, it is yet to be confirmed. But yes, in fact, I emphasize it time and again that whatever we do in our laboratory, not just the culturing, even receiving a semen sample from a person, it starts affecting, or it starts imparting, you know, epigenetic effects. So we have to be very careful and very meticulous in whatever we do.
So, Vasundara ma'am has asked, the composition of culture media is standardized across brands. So, how do we choose which is best for us? And also, what validation steps should we take when we are changing the media lots or changing the vendors? I think,
Okay, so half of this probably I have answered.
yes, sir.
So the other half is, okay, so probably like what I do in my labs, or let me tell you this instance when I shifted from the sequential culture to single stage. Um, as I mentioned, there are different other aspects also why we shifted from sequential. One is the workload, and one is the that I started working as freelancer. So uh, most of the time you do not have a luxury of having help who can change the culture medium and those things. And those laboratories who are using the time lapse, so they do not want to interrupt it. Um, so what you can do is do not jump directly changing your culture medium. If you are using one particular brand and you want to try another, do a sibling oocyte study. So maybe if you have enough oocytes, you can say, for example, you have 15 oocytes, culture 10 in your established culture system, remaining 5 in the new one, and see the difference. Sometimes it's not very evident, and it is very difficult to say just on the basis of culturing one or two patients' uh, embryos. So give enough time and enough embryos to make any conclusion. And if you are happy with the whatever you are using, keep on using that. If you see that the other one is performing better, then you can change the thing. So it's not, I mean, a straightforward validation process, but it requires little patience, little patience, and uh, some uh, such couples which have supernumerary eggs, so you can split those things.
So not to mention about the company names, no conflict of interest here. But for example, sometimes, we go through literature and a lot of literature is available on, Sage Culture Media, for example. And we are using Vitromed, and Vitromed is giving us good blastulation, but we are not having much literature or RCTs on Vitromed Culture Media. So I think, there is no point just because it is Sage and literature. If it's Vitromed is working, we should use Vitromed. What is your take on that, sir?
So, I see any culture medium as a culture medium and do not see the label, because there are many other things. The availability of medium in your area. So now we are fortunate that India has grown so much that even in the remote areas, you can order and you can get any medium which is available in the market today. But there were days when we do not have such luxury. So you can use whatever you want and you can try whatever you want, but then there should be a scientific thinking behind this. So just do not expect any miracle. There are, we all know, those who are working in the field for long, that there are uh, sometimes you can see very good blastulation rate, but still the uh, pregnancy rates, the clinical pregnancy rates, and the miscarriage rates are different. So there, there was uh, one instance when in a particular medium, people started seeing very fine, this phenomenon is known as pitting in the embryos, especially in the cleaving stage embryos. So they thought that, oh, there is something wrong. But then they realized that this medium is giving good results. So not everything is very, bad, and not everything which looks very, uh, you know, as we say, not that all glitters is gold. So it's not always true that you are getting good embryos. I mean, people have tried to correlate the euploidy rates with the different brands of media, and they differ. But again, euploidy is not. I mean, for instance, anything in IVF laboratory cannot be attributed to a single thing. I always say to my students or my friends that, because this was told to me by my supervisor during the PhD program, that the quality control starts with you. So if I'm not well aware of what am I doing, I may introduce, abnormalities in the embryos unknowingly. So even the same medium, um, same laboratory, and the euploidy rates may be different. And for that matter, even the genetic labs that are analyzing your embryos, uh, your biopsies, may not be good enough. So they may be producing false results. So it's very, double-edged sword to say those things. But again, if you are happy with the particular brand, and so please stick to that. This is my advice.
Yes. There's a comment saying, thank you for the excellent and insightful lecture, sir. And the question is, with the growing number of complications reported in IVF-born babies, which are often linked to potential epigenetic alterations, nowadays we see that many companies are rapidly introducing new culture media, transfer media, and clinics start using them in patients soon after their few published papers without long-term evidence or understanding of the possible effects. Considering that even a single drug takes 25 to 30 years of research before clinical use, do you think the IVF field should be more cautious before implementing new culture media or technologies in clinical practice?
Unfortunately, we do not have 25 years to wait. And unless we use them, and this is, as a uh, eternal question, not just for the culture medium, but for any other thing. For example, the cryopreservation. We do not know enough about the well-being of the people who were born uh, through the cryopreserved embryos that were frozen with the slow freezing. And then the advent of vitrification. It has revolutionized. So, and now we have the fast vitrification and fast warming and ultra fast warming. Now we have the uh, publications and studies where they are using simple culture medium to thaw. So the field is rapidly progressing, and you cannot just wait for the um, results to come up. Similar questions arises for the DNA fragmentation. So again, if you do not do this, you will not have enough data. If you do not have enough data, you cannot implement it. And you just can't wait for this. So in a nutshell, I would say the human reproductive medicine is still in its infancy. And whatever based we know, whatever based information that we are getting from the animal sciences, we will have to follow. There is no other.
More than anything, as you say, more than all these things, we should think about fresh embryo transfers. Why are we freezing embryos and creating more and more problems? We really don't know. So I
Yeah.
think those
So,
things have to be given a food for thought.
this doesn't happen in nature.
Absolutely.
Nature doesn't freeze anything.
Absolutely.
So we try to do whatever best as it happens in the nature. But of course, we need to know more about the nature, what it does.
Yes, sir. So how often should we check the pH of the media before culturing? Is it mandatory to do this at every batch we open? And suppose we don't have a pH meter, then what is the protocol we follow?
So if you don't have a pH meter, the question doesn't arise. Okay, so you should have a pH meter. Again, there is a problem with every instrument. Not every instrument is perfect. So, long back I had presented one uh, paper in ISAR. I do not know whether I have that picture in my computer. So it was regarding the temperature. We had a thermometer supplied by one vendor, and it was validated in Pune's laboratory, the National Physical Laboratory or National Electronics Laboratory, something. And the readings from two different thermometers were different. So now you are in a fix whether to believe your incubator's display, whether to believe this thermometer or the another one. And now you have an electronic thermometer specifically marketed for IVF use. So everything that you do and you know, so it becomes more complicated. So keep it in simple. If you have a good instrument, for example, pH meter, do it regularly. Optimum thing is to analyze the pH whenever you have a new batch of medium. More than that, you know, I would recommend all the juniors to read or to do a little homework on the Henderson-Hasselbalch equation. It gives you an idea and why the incubators or why the older books or literature uh, describe about the percentage of CO2. Because we know that certain percentage of CO2 at the sea level or whatever altitude you are at, they are specifically designed to produce this pH in their medium. So read the product insert of that particular brand of medium. It is mandatory because those people, they have optimized their media for that particular, mostly sea level. They describe about the sea level. So if you are having a laboratory at a different altitude, then you will have to do a homework and analyze the pH and everything, because same CO2 percentage will not give you the desired pH there. But if you are working in a laboratory which is at sea level and you have your uh, you know earlier we used to use this Firite kit. The Firite kit was basically used in uh, food industry, and we have adopted from there. And this Firite kit used the potassium hydroxide in this. It was, the instrument looked like hourglass, and you sample the CO2 from the incubator, and then the KOH will absorb the CO2 and it will give you the concentration. So that was enough. For years we have done this. Similarly, so if you have, I mean, it is a surrogate marker of pH, the CO2 percentage, but it is good enough if your laboratory is doing well and if you are observing or monitoring the CO2 percentage. Now we have these, electronic CO2 readers also. But if you suspect that no, the pH is not what I desire, then if you have the luxury of having a good brand of pH meter, you should uh, check the pH um, as often as you need it. These new pH sensors, they can monitor continuously. But every instrument, as I mentioned earlier, comes with its own limitations. Remember, every pH meter probe has a shelf life. So almost every year you need to change it, and it costs heavily. I had a particular brand of pH meter which was specifically designed for droplet uh, measurement, and it cost something around 1 lakh. So every year, if you are uh, purchasing a new probe, it is going to cost you heavily. And if you don't have that much workload in your laboratory, your boss is going to fire you. So uh, everything, even the other sensors, continuous sensors, they have a shelf life. So you will have to balance whether those things can help you or not. Better you ask the uh, company people to come and do the validation for you, but with a word of caution.
Sanjay, are there any other questions there?
I think the last question is single step uninterrupted versus interrupted culture media, which is preferred according to you? So in single step media, whether you'd like to change over or not.
Okay. So more than that, I would see what volume of medium am I using. There are people who use, very tiny amount, 5 to 10 microliters. For example, if you are doing your cultures in time lapse incubators. So they have specifically designed dishes or coins or those things, disposables. So if you see those and if you see the results, it makes no sense to change the culture media. Even they are utilizing very little amount of medium. But and this initiates another debate, because some of them, they are not humidified, and some scientists, they are hardcore advocates of using humidified chambers for your culture. So it's very difficult to say, but uh, if I share my experience, I have done everything. I used large volume. So now what I do in one laboratory, where uh, my junior prepares the dish, I ask her to prepare the culture dish in a center well. So I have at least 1 ml of medium in this, because I do not know whether she is preparing it on a heated surface, whether the uh, time taken to prepare the dish is, you know, minimum or not. So all those things make a difference. It's more than uninterrupted versus interrupted than the change in osmolality and pH. So if your, you know, um, workstation is um, ergonomic and you do I have seen pictures. In fact, I have downloaded those pictures, because these days the social media is flooded with pictures and posts from juniors, and they very proudly show that they are preparing 25 dishes and they are arranged in a very nice manner, making a rangoli of those things. It's dangerous, because more the time you take to prepare the dishes, more you are prone to um, introduce osmolality changes in your dishes. So be patient, do one by one, and if you have uh, some friend or colleague to help you, you can do one person preparing the droplets and the another one is pouring the oil over it. So uh, of course, if you are using too little amount, um, you can change the culture dish maybe on day 3 or day 2. But again, if you see no difference in your, because there are studies which say there's no difference in the quality of embryos or the number of blastocysts. So I believe it is personal, experience and skills, more than saying interrupted or uninterrupted.
So as you were saying about this thing, in extension to this, I would want to know, how do you decide on a single droplet culture or a group culture? And how does oil overlay really have an impact on, culture media? Like, and what is the drop volume normally you use, which will be ideal?
Okay. So earlier I started, because in the early 2006 7, I used to culture them in uh, single droplets, single culture medium, and the volume of those droplets was something around 10 to 15 microliters. Uh, in this particular clinic, we did not have uh, a large volume of work, so mostly 1 or 2 cases. So it takes hardly a few, or maybe uh, half a minute to prepare one dish. I always used oil, uh, except a few instances when we did not have enough time to equilibrate. Otherwise, um, evening before the OPU. And because you are using smaller volume, so there is no chance that you can keep the dishes open. It is only possible with the single, the center well or multi well dishes. But then you will need larger volumes. And the optimum condition, humidity condition in your um, bench tops are not met the criteria of the recommended uh, humidity. Dr. Gabor Vata has done a lot of experiments on this, and he in fact sent me a few dishes and also the um, humidity meter, and I also did the same thing, and I observed that if I cultured, if I kept the medium uh, in a center well, and in the moat I placed the simple distilled water, the humidity was maintained up to 5 days or 6 days even. But we cannot say the same thing with the micro droplets, because even despite having uh, oil overlay, there is a osmolality change due because you are heating them, you are putting them in the cultured incubators. So the osmolality change is profound if you are using smaller volumes. So then I started doing, because when I realized that the other people or the juniors, they might not be that efficient in preparing the dishes, so it's better to use larger volumes. So I'm still using oil, but then I use the center well dishes with larger volume. Oil has its own disadvantages also, because it not only prevents infections or the evaporation and the other things, but also it becomes a sink for the VOCs. So again, and also if you are doing it in a hurried way, for example, you have a um, OPU at say 10 o'clock, and if you do not have the dishes prepared, you think that the medium or the oil kept in the incubator will suffice. No, the moment you put the pipette, the oil from the equilibrated uh, oil vial, it loses the gas. So it is not the same, and you just believe that it is equilibrated, so it will do the thing. So it is, again, um, you will have to balance. You if you want to and of course there are studies that suggest that there is no difference between light oil and heavy oil, so whatever is available you can use.
Thank you, sir. Thank you so much for your time this
uh,
evening on a weekday evening.
Thank
And
you.
I think all of us have benefited a lot. And I think there are a lot more questions, but I request you all to please post the questions on the Scale WhatsApp group so we can convey it to sir and then we'll get the answers from you. I don't think we can hold sir any more longer. Thank you so much, sir. We're
Thank you
truly
very
humbled
much.
So
and
it was really a great honor to have you. Thank you so much, sir. Thanks
I'm
all the delegates. Yes, sir.
I'm humbled to interact with the um, people like you and Sindhuja, who uh, yourselves are very eminent scientists and still waited to listen to me.
Thank you so much, sir. Pleasure always. Thanks for accepting, and thanks all the delegates for being with us today. So,
good
Sindhuja, can you
night,
share
sir.
the WhatsApp link once more in the group? Somebody's asked. Thank
Yeah. Okay, I'll do that.
Take care. Bye bye.
Bye, sir. Good night, sir. Thank you.